this is Walter Schneider again. I'm now describing the data that we collected in the competition. The data, we divided the data into data that we made publicly available and data that we used for scoring. The white in this slide is the data that was made publicly available. That included one development subject, all the data within that process. We also had two additional subjects where we released the raw data but not the validation data. In terms of the MRI data, we had structural diffusion and functional imaging and resting state. That included retinal topology, retinotomy, our language network, the cognitive control network, and the motor system. In terms of the process data, we provided gray-white mass and deterministic tractography and human coding of the major fiber tracks and data sets. Let me review the connectivity data and the scoring. We provided structural imaging, high resolution MP range images, diffusion imaging, where we had both a hardy or high angular resolution 256 direction single shell model, and a diffusion spectrum imaging multi-shell model of 515 directions within that set. So both of these, the two dominant forms of imaging, were run together. We also did the work in terms of all subjects were measured in terms of their resting state, in terms of the functional MRI. And we released for subject 1, but ran on all subjects, the 3 retinal field maps for both vision and for auditory systems, motor system mapping, language mapping in terms of Wernicke's and Broca's areas, and the cognitive control net. We use multiple converging metrics. It's important to note that there is no gold standard for any model, including animal models, but there is a substantial data set of results of what it would be expectations within that set. And we use the known features, for example, the major boundaries between V1 and V2, which are widely replicated across animals, to provide tests to see how well the various mappings are done. Major fiber tracks such as the corticospinal track, which are widely replicated, and known topology of the basic systems, expecting to be reliable both within and close similarity between subjects. We then presented in the morning workshop the details of the presentations. What I'm going to do is continue a bit more in terms of the challenges. So the first challenge was cortical area mapping, then cortical tract segmentation between subject registration and finding tracks known to exist. The metric 1 was the overlap test. The challenge was to segment the V1, V2 borders. This illustrates data where we did 5 replications within subject to identify the V1, V2 borders. The distinctions between the blue and the yellow-orange illustrate the uncertainty of the mapping. And we found for most of the area we could find the border within a single voxel or 2 millimeters of uncertainty as to the location of the maps. Based on that, we can compare these boundaries to what were the submitted boundaries, giving a score of the overlap, the extent to which someone matched that data. So we used a 3-state coding method where we identified what was the hit area for V1 versus the error area, in this case what would be V2 within that. We also had areas where it was ambiguous as to what, whether it was V1 or V2, and did not score that either positively or negatively. So on the right you see what the coded data, every time you had an overlap in the green, you got positive points. Every time you overlapped in the red, you got negative points, and no count for the yellow. So in In this case, we would have an example of a 94% hit rate and 6% miss rate in the border. The ambiguous regions were not scored, and then a false alarm rate if you had reached into that area, which would give you a score, in this case, of your hit minus the false alarm area in terms of the percentage. We then would score them, and for V1 you see the map on the left, and V2 you see the map on the right, where the green area has now shifted for V2 dorsal. If we did not have areas in the near neighborhood, we took a area of uncertainty. That is, if you were 0.05, that would be uncertain. If it was less than 0.001, that was the green area, and the rest of the brain we would treat as negative for that area. For metric 2, we looked at fiber volume track scoring. The challenge was to do the segmentation. And we used a similar map to the three-state scoring operations, where we had known green for known fiber track based on the source to destination. Red was known other fiber tracks, which if you hit that, it would be negative, and yellow, the tracks were ambiguous. And this now shows you an illustration of the coding of the tracks for examining whether or not you got hit or false alarms within that set. For metric 3, we did between subject alignment in terms of the challenge of co-aligning brain 1, brain 0, brain 1, and 2 for each of the subjects. We then did between subject co-alignment. The idea is, could you get, for example, V1 on both the left and right side within subject, and then can you do it between subject? For metric 4, we would find tracks known to exist but beyond the level of current methods. So we took some examples, for example, the optic radiation and the auditory pathway, and asked people, where we identified where the source to destinations are, could they find those fiber paths within that set? We provided resources in terms of links to papers and software, detailed data on the multiple formats in terms of MATLAB and Python, coding routines, and simple programs for challenges 1 to 3. These were hard challenges. Groups had relatively little time, only 2 months looking at the data. The data was complex and in multiple formats. We had 2 strong entries that we thought were deserving of recognition in Challenge 1, and honorable mention in Challenge 2. Only had 1 entry on Challenge 3, and Challenge 4 we had a strong entry, which we also will be giving a prize to. Briefly, in terms of the problems, there was very limited time. We had some difficulties on the alignment of mask, and we had some difficulties on some of the subjects in terms of the identification of the areas between the subjects. This is an example of a score sheet that subjects got of what their score was for executive, language, motor, visual, in all areas, so they could identify which was the best, for example, in motor, or the best in the other areas for that subject. I note that if we take the 2 top entries, entry 1 and entry 3, they really were quite different, and this is illustrated by language, where entry 1 did quite poorly on language, and entry 3 did pretty reasonably on that set. So that there are, different techniques may be better in different areas of the cortex, and the point of the competition is for people to see the relative ranking of the different techniques. This is an example of the match for V1 for entry 3. On the left you can see the green area as it was scored. On the right you can see what the entry did within that process and the mass scoring volume. I note, before we get into the presentations of the others, that we will be continuing these challenges. We'll be doing a IEEE data mining competition, which will begin in August of this year. So please view the winning presentations as well. Thank you.